Hello, and welcome to the second installment of our patient case studies for Bio 252. Let's jump right in. JV is a 68-year-old woman presenting to the emergency room complaining of terrible night sweats and a general feeling of malaise. Upon taking a history, the triage nurse finds out that JV has lost roughly 15 pounds in the last 6 months. JV denies any change in her diet or exercise routine, nor has she begun taking any new medications or drugs which might explain this weight loss. Her vitals are relatively normal, with a blood pressure of 112/71, respirations of 18, a heart rate of 73, and an O2 sat of 99%. She does, however, have a slight fever at 38.5 degrees Celsius. After her blood tests come back negative for an infection, the ER nurse admits her into the medical unit overnight for more tests. You are an attending physician meeting JV for the first time. 3 pieces of information stand out to you about JV: her night sweats, her fever, and her inexplicable weight loss. These are known as B symptoms. The B stands for B cell, and the B cell relates to lymphoma, which is typically a cancer of the B cells. And JV's age, unfortunately, puts her at a particularly high risk for this type of cancer. You run a CT scan and identify an enlarged lymph node in her duodenum. After taking a biopsy of the lymph node, you send the sample to the hematopathologist for diagnosis. The hematopathologist prepares a stain of CD19, CD20, and CD22 antibodies. These antibodies bind the respective proteins, which are expressed exclusively on B cells, allowing the hematopathologist to observe the number and the distribution of B cells in the sample. She notices a diffuse tissue architecture and an increased B cell count, leading to JV's diagnosis of diffuse large B cell lymphoma. JV's healthcare is handed over to the oncology unit, and she is put on the R-CHOP protocol. This consists of 3 chemotherapeutics: cyclophosphamide, hydroxydonorubicin, and oncovin, prednisone, which is a steroid anti-inflammatory, and rituximab, which is a monoclonal antibody. Fortunately, JV's tumor was caught early, and she responds well to the treatment. JV goes into remission after 6 months on her treatment protocol. So we know about JV's backstory. We've learned about lymphoma a little bit, but why did this occur in the first place? So the 2-hit model of tumorigenesis is used to explain the development of cancer. This 2-hit model consists of 2 distinct mutations that have to occur in order for cancer to arise. The first one is a mutation in a proto-oncogene. Once the proto-oncogene is mutated, it is known as an oncogene. So what is a proto-oncogene? Proto-oncogenes are normal genes that we have in our body that are necessary for survival. They control cellular growth and proliferation. So an example here, so the figure on the left is JV's genomic profile. We can see that PI3 kinase, which is an example of a proto-oncogene, when in a wild-type cell, is going to respond to growth factors such as insulin or insulin-like growth factor, and it's going to work to regulate cellular growth, metabolism, and cellular proliferation. Unfortunately, JV has acquired a mutation in this PTEN protein. This PTEN normally works to downregulate or to negatively regulate PI3 kinase, but in JV's case, it's a loss of function mutation, so her PTEN doesn't do that, and her PI3 kinase pathway is constitutively active. This means that JV's B cells are going to be able to grow without regulation by these growth factors, which is not a good thing. The second hit that is required for tumorigenesis is a mutation in a tumor suppressor gene. So like the name suggests, these genes work to suppress tumors. An example here is the BCL6 protein. So assuming that the PTEN mutation occurred first, ideally what would have happened is that the BCL6 protein, through a bunch of different protein mediators, would have sensed that original mutation. And then it would have deactivated a protein, which in turn deactivates apoptosis. And so what that means is BCL6 would have activated apoptosis. Unfortunately, in JV's case, she has also acquired this second hit, which means that her BCL6 protein is unable to inhibit this BCL2 protein. And this means that her B cells are not going to induce apoptosis in response to genetic aberrations. This means that her B cells not only are proliferating uncontrollably, but they're able to accumulate able to more readily accumulate genetic hits and aberrations, which are going to further allow them to grow more rapidly or to adapt to their surroundings. And we can see numerous examples of these, such as with the CD56, the HLA, and these other proteins that are regulated in epigenetic pathways. So what I think is really cool about this figure is it's a genomic profile, which means that JV's, the genetic basis of JV's cancer was determined. And this is really cool because it's a very, very new realm of medicine called personalized medicine, where doctors are able to use people's genomes and a lot of very personalized information to find a really specific treatment protocol that is going to be a lot better than these broader treatment protocols that we would have put JV on, say, 10 or 20 years ago. So I'd like to highlight some things about these chemotherapeutics. So this CHOP protocol minus the R consists of 3 chemotherapeutics, and then prednisone, which is an anti-inflammatory, which is going to work to just help mediate the side effects from these chemotherapeutics. And so what's really important to note here is these are all going to either inhibit DNA synthesis, or oncovin is going to inhibit mitosis by preventing tubulin, which is a component of those microtubules that pull our chromosomes apart during mitosis. It's going to inhibit their formation. I say this because these aren't specific. So these aren't inherently going to target cancer cells. They're going to target all cells. All of our cells in our body are going to take up these drugs. Fortunately, rapidly growing and dividing cells will take up these drugs more readily than other cells. But unfortunately, cancer is not the only example of rapidly dividing cells in our body. Our hairs, for instance, are constantly growing, and that's why we see patients undergoing chemotherapy losing their hairs, because they're rapidly targeted by these drugs. We also see that patients undergoing chemotherapy have GI issues, because their epithelial cells are constantly undergoing proliferation and growth because they're undergoing so much mechanical stress. And so these drugs are going to target those cells too, not just the cancer. Rituximab, on the other hand, is very specific. It is a monoclonal antibody. And if you remember anything about monoclonal antibodies, they target one antigen at one specific epitope, and they are very, very specific. They're not going to target anything else except that one epitope. So what does rituximab target? It targets CD20, which, if you remember, our hemopathology stain is expressed exclusively on B cells. So that means rituximab is going to only target our B cells. And what's more, CD20 is highly upregulated on cancerous B cells. So rituximab is going to have some minor specificity towards cancerous B cells. It's going to mediate its toxicity through activation of the complement system, and it's also going to attract cytotoxic cells and phagocytic cells such as macrophages, neutrophils, T cells, natural killer cells, the whole like. Additionally, it's going to increase MHC2 expression. And this is important because a lot of cancers like to downregulate MHC2 expression. So if we see HLA1 is just a type of MHC2 receptor, and we can see that in JV's case, her cancer has downregulated this MHC2 receptor, and so it's less responsive to TCR or to T cell mediated cytotoxicity. And that's important because it helps the cancer survive. So cancer cells that are readily expressing MH2 complexes are going to eventually present neoantigens, which are antigens that are expressed by the cancer cells. So like this P10, this P10 would be an example of a neoantigen. At some point, these cancer cells are going to express those neoantigens and be destroyed cytotoxically by the T cells. But those cancer cells that do not express those neoantigens by downregulating MHC2 are going to survive preferentially. And so it's kind of like this evolutionary lens through looking at cancer, by which cancers are going to develop different adaptations that help them survive the immune response and help them survive all different conditions that make it hard for them to continue to proliferate and grow inside the body. So we learned a lot about cancer and a lot about lymphoma, but what I really want to highlight here is the trend of medicine from being really broad and going to a much more specific, just becoming more specific and more personalized. Immunotherapies are a great example of this because they depend on antibodies, which, as we know from our immunology lessons, are very specific. They don't target anything except for their cognate antigen. So I've listed a couple other examples of really awesome cancer immunotherapies here. We have our CAR T cells, which Dr. Ott discussed earlier in the semester. And basically these T cells, their T cell receptors are modified to recognize neoantigens expressed on cancers. And then the T cells are reinfused into the patient. There's also checkpoint blockades. So another way that cancers evade the immune response is by secreting anti-inflammatory and immunosuppressive molecules such as PD-1, CTLA-4, TIM-3, and a bunch of others that work to tell T cells to stop functioning, basically, and stop being cytotoxic. And so these checkpoint blockades are actually literally just antibodies that bind those, the TIM-3, the PD-1, the CTLA-4, and these different immunosuppressive molecules secreted by cancer, allowing the immune response to be more effective in the tumor microenvironment. And then finally, cancer vaccines are literally just vaccines that are developed from neoantigens on cancers. And so this is a really good example of personalized medicine insofar as someone's genome is used to develop this vaccine that is specific for their cancer, and that primes the immune system to recognize their cancer and the neoantigens that it expresses. So I hope this is helpful. I hope this is informative. I had a lot of fun making this. If you have any questions about cancer immunotherapy or any of this stuff, I'd be happy to discuss it more, or I'm sure Dr. Ott would as well. So hope you guys have a great rest of your semester, and peace out.